So I want to start out by reminding everyone that the SolveCFS Initiative, formerly known as the CFS Association of America, has had a very long and successful history of funding ME/CFS research. We actually started in 1987 funding research, and up to today have provided research funding for some of today's best and most expert ME/CFS clinicians and researchers. Over the years, our funding has focused on specific topics, from virology and immunology to neuroimaging, endocrinology, post-exertional malaise, and education. And then in 2008, we began a competitive funding process where we issued requests for applications, and then we received a number of applications, and we would make awards, grant awards, based on competitive review and peer review of these grant applications. These competitive funding opportunities in 2008 and in 2011 focused on biomarker discovery, biomarker validation, and treatment. So rather than specific topics, research topics, we opened it up and really allowed it to be investigator-initiated funding opportunities. Still, despite the many significant and important research results that have occurred over the years since 1987 and are now in the published scientific literature, we as an organization and the ME/CFS field in general really wrestled with the fact that researchers did not have the opportunity to collaborate and share. And this is really analogous to the blind man and the elephant parable, where we as ME/CFS researchers were all examining different parts of the elephant, or ME/CFS, if you will, comparing results, and yet not agreeing on what we were describing, what we were researching, and what we were defining. So in 2008, with our first competitive funding opportunity, we awarded 6 grants to these investigators. Gordon Broderick, who was then at the University of Alberta in Canada, Sanjay Shukla at the Marshfield Medical Research Foundation, Kathy Light at the University of Utah, Bud Mishra at NYU, Marvin Meadow at New York Medical College, and Dikoma Shangu at Weill Cornell Medical College. All these investigators agreed to use similar measures, share their data, and collaborate. This was the first ME/CFS research network. And we had a number of successes. We engaged 3 new investigators in this 2008 funding opportunity, new to ME/CFS research, that is. Projects were both hypothesis testing. For example, they used imaging to look for new markers of ME/CFS. There was hypothesis testing about the autonomic nervous system and biomarkers, and also hypothesis generating, like looking at the microbiome and what implications does that have for ME/CFS, and generating a knowledge base of all the ME/CFS experimental data and published literature. Importantly, this 2008 funding opportunity, which was provided seed funding to these investigators, led to follow-on funding of $7.5 million for several of the investigators. And also, many of the collaborations that were established from this research network continued today. We also learned some lessons from our 2008 funding opportunity. We learned that the lack of access to well-characterized patients and their samples really prevented some of the research from moving forward. We also learned that lack of infrastructure prevented investigators from sharing and also archiving the data. in a way that was retrievable and usable by others. And we also learned that seed funding was insufficient to make ME/CFS a focus of the investigators' research portfolio. So clearly there were barriers for some of our researchers and for progress to be made in ME/CFS research. To lower these barriers to ME/CFS research, we created the Research Institute Without Walls in 2010. This infrastructure, and I say infrastructure in quotes because there is really no bricks and mortar to this infrastructure. It's really a system set up to give the opportunity for patients to participate in research, for investigators to participate in research in a way that was very innovative and cutting edge. Now we have the SolveCFS Biobank at the center of research. This gives, again, patients the ability to participate in research from wherever they are in the world. The SolveCFS Biobank also provides samples and health information for researchers. And now the Solve ME/CFS Initiative is a partner in the research process rather than just an organization giving gifts to researchers. And the Research Institute Without Walls enables collaborations and partnerships. And we also use the Research Institute Without Walls and the SolveCFS Biobank to help recruit patients for clinical trials and also for other research studies that are independent from ours. So it provides a very robust system and infrastructure to accelerate the research process and lower barriers to research. Our 2011 competitive funding opportunity was the first that used our Research Institute Without Walls. We made awards to these 5 investigators. We have Dane Cook at the University of Wisconsin in Madison, Peter Rowe at Johns Hopkins Medical Center. We have Marvin Meadow at New York Medical College, Patrick McGowan at the University of Toronto, and Spiros Defterios with BioVista, a small biotech firm. 2 of these researchers are also new to ME/CFS research. Now, the Research Institute Without Walls puts the Solve ME/CFS Initiative right at the center of the research process. We are now the research hub. Investigators use samples, as did Patrick McGowan and Spiros Defterios, from the SolveCFS Biobank. They use the samples or the health information provided by our biobank participants, or the investigators let their study participants know about the SolveCFS Biobank so that they can sign up. Importantly, every investigator now archives their research results with the SolveCFS Biobank using the REDCap system, which is the Research Electronic Data Capture System. So all of the research that we support, that we fund, is now permanently archived with the Solve ME/CFS Initiative. Since 2011, this research funding has already resulted in $1.5 million of follow-on funding for 2 of these investigators. BioVista is in the process of raising funds to conduct a clinical trial as a result of their findings from their research project. And the other investigators are using the data from their funding to apply for follow-on funding. The SolveCFS Biobank, again, at the center of our Research Institute Without Walls, has successfully lowered barriers to ME/CFS research. In less than 3 years, 9 of the brightest investigators from some of the best institutions are conducting ME/CFS research because of the SolveCFS Biobank. And here they are. Mike Cooperstock from the University of Missouri started ME/CFS research using our SolveCFS Biobank 2 years ago. Eric Delward from the University of California, San Francisco, and the Blood Systems Research Institute also used samples from the SolveCFS Biobank to conduct ME/CFS research. Steve Elledge, a Howard Hughes Medical Institute investigator from Harvard University, is using samples from the biobank. Durya Unitmaz from New York University Langone Medical Center is studying ME/CFS using the biobank. Armin Aladini from Columbia University. Michael Houghton, Lasker laureate, now at the University of Alberta, is using biobank samples for ME/CFS research. And Patrick McGowan and Spiros Defterios, also grantees from our 2011 funding opportunity using the biobank. So again, pretty darn impressive that in less than 3 years, 3.5 years, we have been able to attract these amazing investigators doing some of the most innovative research on ME/CFS into ME/CFS research. The biobank is also used to conduct what is known as clinically meaningful research. What is important to the patient about their disease? That is what is meant by clinically meaningful. And we use that is possible because of the research electronic data capture system of the SolveCFS Biobank, where patients are able to record information about their symptoms, their experiences, and the impact of ME/CFS on their lives. Lenny Jason from DePaul University is using this health information to learn more and work on issues about ME/CFS case definition. Shelley Wang from MD Anderson Cancer Center, a new investigator to ME/CFS research, is working, and she's also an expert on symptom research, is using her expertise and the SolveCFS Biobank to come up with patient-reported outcome measures for ME/CFS research. Again, something that's critical for us to be able to move forward with getting FDA-approved treatments for ME/CFS. And Spiros Defterios used information from the SolveCFS Biobank about medication use and medical history for their drug repurposing project. So the Research Institute Without Walls has effectively brought the elephant into the room, and we've turned on the lights. Now researchers can collaborate and share if they want to. They can also continue to focus on their specific area of research. But the important thing is now all of the research is archived, and we have the capability of integrating all of this data together. The SolveCFS Biobank puts patients at the center of ME/CFS research. Everything in the same place. Now I'd like to switch gears just a little bit to tell you more about the way the SolveCFS Biobank works. And there's been a number of questions over the past couple of years. And so hopefully this information will help bring some clarity to how we've designed this very innovative structure. One of the reasons that ME/CFS is challenging to research is because there is no lesion. There's not something that a doctor can go and feel or measure or take a biopsy of. So we've relied, we as a research field and we in the SolveCFS Biobank have relied on the use of blood. And this is important because blood is a great sample. It travels. There's not a place in the body where blood doesn't go. And in this journey throughout the body, blood can pick up signals and molecules along the way that will give us clues about the possible causes of CFS, about diagnostic markers, and other things that again will help us figure it out. It's a great sample because there are a number of components in blood. You have the liquid part of the blood, or the plasma, which carries a lot of molecules. You have cellular components like the white blood cells and the platelets. All of these, again, can be used to give us clues about the causes and biomarkers of ME/CFS. We've used blood and blood from in our existing biobank inventory to conduct and fund and support the cutting edge research that I just showed you the investigators are doing. Well, I didn't show you the research, but I showed you the investigators. Here's some of the research, the kinds of research that's being done by these investigators. They're looking for novel and previously characterized viruses in the plasma. Again, that's the liquid part of the blood. And viruses end up in that part of the blood if they leave the cell and they circulate throughout the body. There's also research being done on novel agents, novel infectious agents in the blood. And not only in the liquid component, but also in the cellular compartment of the blood. Research is also being done on the history of virus infection, using antibodies to create a fingerprint, if you will, of virus infection throughout the person's life. We're also using the biobank blood samples to look at autoantibody profiles. This is the work that's being done by Steve Elledge at Harvard. And we're looking at the cellular component. How do the immune cells look? What is their signature? How are they different in CFS compared to healthy controls? We're looking at the epigenetics of CFS and the epigenetic profiles in the blood cells. We're also looking at how these epigenetic patterns affect the function of the blood cells. And finally, this blood sample is also being used to validate diagnostic markers that have been identified and now require validation in order to move forward as an objective diagnostic biomarker. Many of these studies are ongoing, and actually many have just started within the past year and a half or so. So most are still ongoing. But the exciting thing is we have some manuscripts that are in preparation, manuscripts that are in press, and very exciting, we have a manuscript on the epigenetics work that's been done by Patrick McGowan that's embargoed. It will be published in the next 2 weeks. The important thing about our Research Institute Without Walls and the research that it supports is that when these research results are ready for prime time, we let you know about it. When they're published, we will announce it so that you will always know what your participation in research means, what contributions you have done to really further the research in ME/CFS and show you how you are, in fact, being a part of the solution by participating in the process. This slide is to try to demonstrate how a little bit of blood, these 3 tubes right here, amount to maybe a small juice glass of blood, goes a very long way in the biobank. And I show this because actually all of the research that I've talked about so far has been a result of the original study that we started with GlaxoSmithKline in 2010 to look at XMRV. And by the way, we just got word that paper will be published in a couple of days. So that will be an exciting explanation and demonstration of the power of patient-centered research and our SolveCFS Biobank. So the blood that was collected was aliquoted into many different sample vials. Remember the different components that I showed you are put into various tubes. Then depending on the type of research that was done, that requires a very specific type of sample, whether it be viruses, whether it be gene expression, whether it be serum for autoantibodies, the right sample goes to the specific type of investigator. Then, once the investigator is done, those research results are logged in with us and all relating back to the original sample, or you, the original participant. So a little bit goes a long way, and this is really the equivalent of an individual participating in many different types of research studies, or if these were clinical trials, many different types of clinical trials. So this illustrates the power and the nimbleness of the SolveCFS Biobank to accelerate research, to lower barriers to research, to integrate information, and to relate that all back to the individual. This is a little bit about the process of how it works. Again, so that's kind of the blood part, or the actual sample part, the research end of it. Now here's where you come in. To date, we have about 1,000 people in the biobank, and the first step for every one of those 1,000 was consent, and we are using a hard copy consent that says you agree to participate and in this study and more, and provide a sample. So once you sign that consent, you make a copy to keep one for yourself, and you mail the original signed copy back to Gloria Smith, our biobank coordinator. When Gloria receives your signed consent, she then sends you an email. And what this email is for is to gather specific types of information. It's actually very basic information. It's your name, your birth date, where you were born, and this information is used to generate what is called a GUID, or a Global Unique Identifier, which is essentially a hash code of your information. This hash code then stays with you forever. This gives us the ability to share your information and your sample, which will have only that hash code attached to it, with many, many researchers. So they do not have any of your private information, only we do. But because of this unique hash code, we are able to then integrate and share that data among investigators safely and securely. This is also a place where many of the 1,000 people that have signed up have been waiting now. You've been signed up, you're registered, your global unique identifier has been generated, and you're waiting for the next thing to happen. And you're standing in the ready. Well, that's about to happen soon. What we now know is that we need to grow the biobank to be as representative of the ME/CFS population as possible. So very soon, all of the participants will be receiving a new email that provides links to surveys that will collect demographic data and also some basic health information. And again, what we will do is we will use this information to determine how representative our biobank population participants are and what populations we might need to target next. Once we have that information, again, your basic demographics and some basic health information, we use that to match you to specific research studies. When you're a match, you are then sent a kit, and that kit might be specific types of blood tubes. They might be purple top blood tubes or red top blood tubes, or it might be a tube that you spit in. Whatever type of research is, we try to meet the requirements, or we don't try. We do meet the requirements of the specific types of research. And again, the sample has to be appropriate and exact for the type of research that's being done, or else the results that are created or generated are just nonsense results. So you will get that sample collection kit with all the instructions in the mail. You then spit or have your blood taken. And for those of you that are not able to use your doctor to collect a blood sample or go to a laboratory like Quest, we have a contract with Exam One that is a mobile phlebotomy service. So they come to your house to take the sample. And we provide you everything you need to then return that sample to the SolveCFS Biobank, usually very quickly, if not within hours, then overnight, to the SolveCFS Biobank, where it's processed so that it can then be sent to the investigator. So this is really what patient-centered research is all about. We've designed the SolveCFS Biobank so that it is a robust research that lowers barriers to really increase the number of high-quality researchers that we have doing ME/CFS research, and you are at the center of this. If you are interested in being at the center of research and really driving ME/CFS research, find out more about the SolveCFS Biobank. See if it's right for you. Contact Gloria. She's our biobank coordinator. Here is the phone number that takes you to our biobank resource line. It will be a message. Leave your name, number, and Gloria will get back to you. The number is 704-362-2343, or send Gloria an email. And here's the email address. Simple. biobank@solvecfs.org. Also, over the coming weeks, we're going to be needing more of your help. If you're signed up already, we're going to give you the tools that you need to help us help you enroll your family and friends. So we need family members in the SolveCFS Biobank because they serve as great controls for specific types of genetic studies that we're gearing up to do. And we also need your friends because in many instances, your friends are very much like you in many ways, and they serve as good, healthy controls. Also, if you have your family members and they are not healthy, if they also have ME/CFS, or if they have another similar condition, we still want those types of family members enrolled. So again, over the next couple of weeks, we'll be giving you the tools you need to help you enroll your family and friends. So be at the center of this solution for solving ME/CFS. We have a number of webinars coming up over the next several months. Each of those will be by investigators that are using the SolveCFS Biobank in one way or another, or that we have funded. So this is a very, very exciting webinar series coming up. You can also be part of the solution by signing up for our Research First newsletter. Remember, all the data, all the research that's generated using our donor dollars for research and using the SolveCFS Biobank will be reported in our Research First newsletter, both our e-newsletter and our hard copy. So stay up to date and really stay involved and know how you are contributing to the solution. Like us on Facebook. That always helps. We lots of likes is a good thing. And our Facebook address is facebook.com/CFIDSASSN. That stands for CFIDS Association. I guess that's one thing we can't change. That's that code is I guess stuck with us now. And of course, last but certainly not least, perhaps most important, drive research and fuel progress by donating. I think I've showed you that we've gotten incredible value from our biobank and from our Research Institute Without Walls. But still, research costs money. And I hope I've convinced you that the research that you are clearly a part of and helping to support is some of the best that's out there. So I'd like to thank you for participating. I'm going to turn off the recording now, and I'll take a minute or two to see whether or not any questions have come up. Again, thank you very much for listening.